So now basically we're looking at different hazard categories, and the idea is that category 1 is the most toxic, and then when the numbers increase it becomes less toxic, and so in the end you want to do a classification and labeling of the chemicals according to the hazard classes on the one hand, but you also want to determine so-called safe limit doses. So you have to understand the derived no or lowest adverse effect levels, acceptable daily intakes, and also acceptable operator exposure. So this is why we're doing all this, at least here in Europe. And so what are the observational studies in animals to derive the factors, so the classification as well as the safety factors? So when you look at the skin, you have a single exposure for example, irritation, but also repeated exposure when you consider skin sensitization, because the immune system has to be primed first. Then for the acute toxicity, you have a 14-day period that you monitor the animals. Then you have repeated dose chronic toxicities, also to determine lethal doses. Then you have lifetime exposures for carcinogenicity. Then you have the organ toxicity that also takes weeks to months. And then you have the reprotoxicity, where you look at the pregnant animals for the reproduction, the development, and then also sometimes the F1 generations. So this is really a lot of different tests that have to be performed, but not all chemicals you have to perform all tests. It depends on the tonnage. So the higher the tonnage, the more information should be derived. And maybe it's not that intuitive to begin with, but the idea is that the higher the tonnage production of a chemical is, you can basically equal this with exposure. So the higher there is the exposure on the individual level, and the more people are exposed. So that's the rationale behind that, although the rest of it then concerns hazard and not exposure anymore, because the tonnage is basically covering this. Well, what is the problem with the current paradigms? So these are all OECD guideline studies, similar to what you have for the ecotox, I guess. The problem is that for all these studies that I just showed you, it's really a logistical problem. So it's impossible to test like more than 30,000 chemicals in our environment with these OECD guideline studies. So they take a lot of time, they're cost intensive, then they also take a lot of space, of course. And for example, just looking at developmental neurotox, so that's my subject, it's really one small endpoint out of a lot of different endpoints. One compound costs more than €1 million or dollars, or I guess even more Swiss francs, and takes more than a year. So that means if you really want to cover the hazard by itself with the standard test, this is actually not possible. And this is one of the reasons why, when you look at the database that we have here of tested chemicals, so it's a little small, but here are the high production volume chemicals are here. You see that there's really very few information, and just 75% of the 2,000 to 3,000 volume chemicals on the market, there is insufficient toxicity, and also not only human but also ecotoxicity data. So it means, so the more complicated, the more labor and resource intensive the testing becomes, of course, the less information we can provide just due to logistics. And this was one of the reasons why in 2007, the National Research Council of the United States actually suggested a paradigm shift in toxicity testing. You're probably all aware of this. It's really the first document that kicked the whole alternative or new approach method field off, because it suggests we have to shift the paradigm from looking at apical endpoints in animals to a mechanistic testing using in vitro human cells, if possible, for the human tox, and also in silico methods, and looking at population-based exposure data as well for interpreting toxicity testing results. So this was in 2007. Now we have 2024, so it's almost 20 years have passed. The field has moved a lot, but in the end, how do we perform chemical testing today? I mean, still the old way. So we're basically in a transition from the old to the new, because meanwhile there are a lot of strategies have occurred to try and get away from the old. But it's really a process that is not only scientifically difficult, but it's also psychologically difficult, because risk assessors also have to they have to go with the flow, they have to go with the science, they have to learn a lot, educate themselves a lot, because all these new methods and these new technologies, and this is actually a very nice article that summarizes that what's called new approach methods really nicely. And I bet a lot of it is similar between the ecotox and the human tox, like computational modeling methods here, high throughput screening, omics applications, advanced imaging and scanning techniques. And then for the human tox, you have the advanced cell culture system, like the 2D and 3D cell-lysed, a lot of induced pluripotent stem cell work from human iPS cells, and also then the microphysiological systems, then organoids, organs on the chip, humans on the chip. I don't know if there are fish on the chip yet. I don't know, but Christine, you probably know better. And so and science is moving so rapidly at the moment. I mean, if you try to keep up with all the publications that are published just every month, it's a lot for a scientist, but it's even more difficult for regulators to keep up with the novelties in technology. But nevertheless, so I would say within the last year, a lot has also moved on the regulatory side. So regulators have become more involved, more open, more in the discussion to try and also adopt these novel technologies. So the US have been, not only because of the 2007 document for the 21st century toxicity testing, but also on the new approach methods work plan. So this is also really from 2014, so it's also already 10 years old, but they have a really nice diagram, I thought, in there for how to get the NAMs, so the new approach methods, into regulatory application. And on the one hand, of course, you need NAM development, and you have to make sure that the NAMs that you develop, that they are valid, so that they are reproducible and that they are predictive and that the biology behind it is correct. So that means behind the development, then you have to establish the scientific confidence of the NAMs. So the NAMs have to be fit for purpose. That is important. So for example, if you want to do like a nuclear receptor screen of 1,000 chemicals, you wouldn't choose the organ on the chip system for that, so the purpose has to be adapted to the question that you ask. And the scientific confidence is really important to demonstrate that the application for regulatory decisions really holds. And as the EPA, they published this, like I said, 10 years ago, but they are not alone anymore. So, I mean, there is ECHA, there is the FDA, there is even ECHA, there is EFSA. So there have been a lot of reports popping up from the agencies, kind of trying to understand how can we develop a roadmap towards the change in regulatory chemical assessment. And now the EU is also working on a roadmap. I have that on, I think, on my last slide, which I think is a really good sign, because in the end, the European Commission, at least for Europe, and I think Switzerland kind of always leans towards what Europe is doing there. In the end, the legislation are made by the European Commission. So when the European Commission has to be involved in the change, because this is something that all the stakeholders have to do together. I'm really convinced that this is very important. So you need to talk with the legal people, but you also have to talk to the regulators who use the laws And you have to streamline this with a test method developer, so that when you develop a test, you already know what the regulator needs to fulfill certain criteria that it can be applied. And I come to this in my case study for developmental neurotox, where we did this from very early on, and I thought it has been really successful to have a very, very good and very tight communication. Okay, so one thing that is really important is the scientific confidence, as I said. And this paper, I brought this because I think it sums it up really nicely. On the left-hand side, you see where the authors are coming from. So the OECD, the National Toxicology Program, but also the Joint Research Center, the EPA, and PETA. And they published this scientific framework for establishing the confidence. And that's probably true not only for the human tox, but for the ecotox, it will be just the same. So like I said, the fitness for purpose is something that needs to be described. Then the biological relevance, might it be for a human or for a fish, or if you go to veterinary medicine for whatever species. Then the technical characterization is crucial, but it also has to have an independent review, and the data needs to be really transparent. And so after giving you now a lot of theory, I will now go a little bit into practice, how we did this for the DNT. So I thought it's nice to understand where my background is, so where I'm coming from, because obviously I'm not in the lab anymore, I'm not doing this anymore, but it's really helpful that I have this understanding that I've been in the lab for 20 years and can now go more into the application side. Okay, for the developmental neurotox, or DNT, this we had actually also in 2014 this international stakeholder network for DNT, and I brought this because it actually took place in Zurich, and it was organized by Martin Wilks from SCAT, so he is here amongst the authors. And this was the very first time that I got in contact with SCAT. Before that, to be honest, I didn't know it even existed. So that was the first contact with Martin, and since then we've been having a really nice scientific relationship. And at this stakeholder network meeting, there were people from regulatory agencies there, so it was mainly EFSA and the US EPA. And we had people from industry there, and then we had academic scientists and the JRC organizing the scientific frame. And we sat around the table for a couple of days and just lined out the roadmap. How are we going to proceed? How are we going to get the new approach methods for DNT on the road and into regulation? And then it took basically 9 years from then to have an OECD document as a recommendation in hand, and I'll guide you through that. Okay, so DNT is kind of complicated, because there are lots of different processes that needs to be integrated when our brain forms. But nevertheless, you can break this down into key neurodevelopmental processes. So it's pretty clear then when the embryo forms that the cells, the neural precursor cells, they have to divide, they have to proliferate. Otherwise, we have a too small brain, we have a microcephaly. Then the cells also, they have to migrate and find their right spots in the brain. And while they migrate, they differentiate in different cell types of the brain. So at first they're radioglia cells, then they become neurons, they become astrocytes and oligodendrocytes, which actually perform the myelination of the neurites here. The neurites need to grow and expand, and in the end, we need the formation of a functional neuronal network. And so these processes are not, of course, not the only ones that have to work out, but they are so basic that a group of experts that formed internationally also many years ago decided, okay, if one of these processes is disturbed, we will have an adverse outcome for the brain. For just one example, lead exposure in children inhibits synaptogenesis, and you have a lower IQ in these children. So similarly, Zika virus infection affects the proliferation of the neural precursor cells. You have a microcephaly, also an adverse outcome in the children. And so this is the principle behind it all. And then actually 3 labs, and that was the US EPA, that was Marcel Leist's lab from the University of Constance, and my lab at the IUF. We developed assays that cover these neurodevelopmental processes. So you basically, for each of these processes, you have at least one assay that you have in an in vitro system in the dish. So we ended up with a battery that has 17 assays right now and covers 8 different neurodevelopmental endpoints. And these assays can be used to test compounds and generate concentration response curves like you are all familiar with. And when you cover the whole battery of tests with a chemical, then you can look which endpoint is the most sensitive, and that would be the endpoint of regulatory concern. Yeah. Okay. Now I'll go a little bit into what the system is that I used to work with. These guys are called Neurospheres here on the left side, and actually these are human neurospheres, but you can also make neurospheres from rat, from mouse, from rabbit. And actually within the PARC project, I don't know if you're aware of this big 400 million EU regulatory project now. It's actually a former postdoc of mine. She's now at the BFR in Germany, and she is trying to make neurospheres from zebrafish. So, you know, you can do it just with whatever species. I mean, for most in our hands, it always worked the same way. And so these guys are proliferating in 3D, but you can also plate them onto a matrix. And this is the former sphere core here. And this is like a 5-day migration. So when you plate the spheres, then the cells radially migrate out of the sphere. They first migrate as radial glia cell, and this is what happens also during cortex development. And after that, the neurons and also oligodendrocytes migrate on top. And here's a little bit close up. You can see that there's a multicellular lawn forming around this sphere. And with this neurosphere has the advantage that it's a high content assay. So with just the proliferating and the differentiating stage here, we cover 7 of the 17 in vitro battery assays and 5 out of 8 endpoints. So it's actually also time and cost efficient. So how do we gain now trust in the in vitro battery assays? And this is exemplified with Neurospheres, but all the other test developers have done more or less similar experiments with their spheres. So for one, of course, you have to show the reproducibility. So I'm not going to show you data that's kind of boring, but you have to do your homework. Show what is the reproducibility of the endpoint specific controls so get basically historical control data where you see how stable your test is. I bet that's this is common sense for test development. Then the relevance for the biological plausibility is important, and the predictivity. So how predictive is it, and where are your applicability domains? And now I will actually go a little bit into the biological relevance. So we've been doing this on different levels. So one level is happening within the Horizon 2020 Entox project that is part of the SBIS cluster. And you might be aware of the Precision Tox project, because this is also dealing with non-human species and kind of merging into the ecotox. And we are one of the partner projects here. And what we've been doing, we've been developing something that's called a physiological map. It's not finished yet, but what you see are all these different brain cell types. And on the right-hand side, also timing, because of course that also plays a role during development, and the different processes. And in the end, so this is supposed to be open source at some point, you can click on, for example, here, you can click on the oligodendrocyte, and that takes you then to the next level of the map. And this is all information from humans that we gathered. So we really are trying to avoid animal data, because especially the brain although there are basic similarities. So, you know, we all have reflexes, we all breathe and so on, but the human brain is different from a rat brain. I mean, just because the goal, what the rat should fulfill in life, is a lot different from what we have been developing during evolution in, like, I think 60 million years, that we actually branched off the rat and the mouse, so the rodent. And so it's mainly the forebrain in humans that then expanded, and, you know, the surface folded, and we have many different tasks that we do comparison to the rat, that basically needs to find food and reproduce as much as they can. So, you know, the goal in life is a little bit different. And this is why we've been trying to really stick to the human species. And this is oligodendrocyte development here. So you have the specification of the cells, proliferation, migration, differentiation, and then the maturation and myelination of the oligodendrocytes, and then all the signaling, what we could find, what relates to these different processes. And in the end, these physiological maps are basically as a, it's like a map where you then can orient your in vitro system in. So which signaling pathways can my system cover and what cannot be covered, for example. So it kind of builds, it's supposed to build confidence and trust. So that to the physiological map. And then we've been involved in another EU project that is coming to an end right now, of the Urian cluster. And that's called Endpoints. And we have been looking for endocrine disruption-related DNT. And so looking into the literature, there wasn't really that much out there on nuclear hormone receptors and the human brain development. So we used our system to also understand what are the tasks of like, we looked at 14 nuclear hormone receptors during brain development. And this is just a summary of the results. And again, in the middle here, you have the neurodevelopmental processes, again, proliferation, migration, differentiation, maturation. And here are all the different hormone receptors that we found out by doing pharmacological inhibition studies or stimulation studies, and actually co-exposures and RNA-seq analysis, just to find out which of these hormone receptors contributes to which process. So in the end, we think that also these set up as test methods can be used for looking at a human relevant, probably relatively sensitive endocrine disruption DNT endpoint. And so there's this French public-private platform, PEPPER. It's a validation platform, and one of our assays that concerns the glucocorticoid and the retinoic acid receptor is under validation with PEPPER at the moment. And this is basically making the assays fit for an OECD application. So this is also about gaining confidence on the endocrine side. And so the third strategy was to come from neurodevelopmental diseases. The rationale behind that is that we're saying, well, if you test in a rat, you will never find an outcome in the rat that is autism or ADHD, because there is nothing like an autistic mouse or rat. There are animals that have certain traits of the human diseases, but they still, it is different. How can you approach this issue now from the in vitro side? Well, there are actually quite some mutations out there. These are usually rare mutations in humans that lead to neurodevelopmental disorders. And so we went through the literature and collected data on which pathways, when they are disturbed, lead to an adverse outcome in humans. And then we again used modulators of these pathways and checked if our system can represent disturbance of these pathways. And these are examples here of, for example, Notch. This Notch triggers a basic switch between neurogenesis and gliogenesis. Then there's also mTOR, COX2, Wnt. I mean, these are all very pretty much conserved pathways. And so we tested all these pathways that are around here on the outer circle. We tested on the different neurodevelopmental endpoints that are here in the core center. And actually all but one were were represented by at least one of these neurodevelopmental key events, which means if a compound acted on a pathway that is known to be pathognomonic for human disease, the assays would pick this up. And so this was another step towards building confidence. Okay, now what about the predictivity? For the predictivity, you have to screen compounds, and this is actually what we did. This was an EFSA project that ended in 2021 already, and this was testing of 120 substances in the constants and the IUF assays. And the US EPA actually also tested the same compounds in the US. And what is very difficult for DNT is to find positive and negative compounds, because there is just not that much information out there, because there is a huge data gap. We just don't know of these 30,000 compounds if some of those interfere with human brain development. So, but there is a small list of 45 that we were pretty confident that these are the positives here, and on the right side that these are the negatives. And this is the evaluation of the compounds just in the EU assays. So the evaluation of both together with the US is still ongoing. And so we have a sensitivity. Whoops, where is it? It disappeared. I'm sorry, my numbers disappeared. I must have deleted them. So I'm sorry. So we have a sensitivity, this should be there, of I think 68%, and a specificity for these compounds here. You see there's only true negative, so there is no false positive, of 100%. With the sensitivity of 68%, we were pretty happy because the EPA assays were missing. When you put them together, then the specificity actually goes a little bit down because there are more false positives on the US side, and the sensitivity goes up. So this is published, so I'm sorry that I miss out the numbers. Okay, and because we had a lot of these data gathered together, in the end now in 2023, this OECD initial recommendations on the evaluation and also data interpretation from the DNT in vitro test battery was published. And that was really a big success. I mean, we would have liked to have it a guidance, and that it in all the preparation time, it was called not a guideline, but a guidance document. But then in the last round of commenting, there was one member state who said, we want to have the title changed. Okay, but the content is still the same, and it's actually a step. So in the guidance document, actually there are a few gaps were identified, of course. I mean, it's not perfect, which concerns the lab-to-lab transfer of all these 17 assays, which had not been taken place due to resources, biological gap closure, and also increasing the database. And lucky enough were we, because EFSA decided to put, again, more money onto the table to close these gaps. And like I said, I think because we were in communication that very early on, so EFSA was involved in creating the roadmap. That's why we were just really lucky that they continued also to fund the idea. And and so it just this project just started now in 2024. And all the assays, all the 17 assays from all 3 labs will now be transferred actually to the Entox. And the Entox was introduced now in the introduction. This is my startup company that I started with the 5 co-founders. And the idea is that these assays are also available for the market, because what good is it when science develops assays, then maybe even a regulation changes, but nobody can buy the tests. I mean, then it would end right here. So it's really important that we get these assays also out onto the market so that they can be used, so that industry can come and say, we really like this, doing this an alternative way and not killing thousands of animals, and we will produce a case study, but therefore they need a partner that they can do this with. And also getting in the end to a GLP level is really important, and you cannot do this out of an academic lab, at least not the labs that that I know of, where all the students jump around and fridges, you know, open, close, tubes not really written on correctly. So that doesn't work. So you really need to get this onto a professional level. Nothing against students, but it's sometimes a little chaotic in the lab. So it's just how it is. Okay, so the lab transfer is will actually happen this year. Then biological gap closure, and that mainly concerns glial cells. EFSA also took some money into their hands, and this is a 3 million euro project now that is led by the Norwegian Institute of Public Health. And what is really nice, University of Lausanne is involved. So there's Marie-Gabrielle Zürich that I've also been collaborating with for many years. She's on board Karolinska Institute, and now also me because of the SCAT. And then increasing the database, it's also happening. And here, the National Toxicology Program in the United States at NIH, they are committed to test more than 300 compounds from 2021 through 2025. So we are gaining more information here. And also for EFSA, the Entox will test 100 pesticides now in the next couple of years to increase the database. And the final goal is, of course, to have an OECD test guideline and have DNT testing mandatory, because right now it is not mandatory. I mean, you don't have to test for that. And I think this is really a no-go. And the agencies like EFSA have also recognized that this is really not a good thing. And also the FDA in the US is now on board. So they have identified, like the EPA, DNT as a major concern. Okay, but in the end, these tests have to be somehow taken into the regulatory frameworks. And of course, that's a challenge, because from the animal experiments I showed you, I mean, you cannot say this is one acute tox study and now I have one in vitro test. That just doesn't work. So, but there are really a lot of people out there who are trying to solve the problem and trying to find ways to make it happen. So first of all, you have to distinguish, do I want to screen and prioritize, or do I really want to do risk assessment? And there are then different strategies involved. So especially for the risk assessment part, it's clear that the NAMs are no standalone tools. I cannot take one in vitro test and say, oh, that's sufficient now. So it really needs a framework around it, and this is the adverse outcome pathway concept. And this is just an example here of an AOP network for Parkinsonian motor deficits that I have been involved in many years ago with EFSA. And so this gives you the idea if it's biologically plausible, if the weight of evidence is enough, and so on. And that's on the hazard side, and on the exposure side, in vitro to in vivo extrapolation we also need. Otherwise, we are not getting anywhere, because we are working with nominal medium concentration that you don't know which concentration in the end triggers the cellular response. So that's probably very similar also in the fish, or maybe sometimes even more extreme, because the water volume, I think it's much bigger than in the cell culture. And so there are concepts for this, what's called now next generation risk assessment. And I brought you just one example, but there are different groups who have published also different papers. But I thought this was really nice. This is also from the ESPIS cluster. So there are certain modules. So ADME, and exposure is one module. Then the hazard identification and characterization is one module. And then you can integrate the whole thing to get also a risk assessment dossier. But in the end, to fulfill the regulation how it is right now, you would have to do this for each of these endpoints that I showed you in the beginning, you know, the slide with the dead rats and stuff. So, I mean, for all these endpoints, you need this information. So that means you have to follow the scheme for each of these endpoints. But that enables you to come up also with hazard classes. And this is taking into consideration increasing potency and increasing exposure. So in the end, it's not that different from the tonnage and the hazard that we are looking at right now. You just have to get used to that the methods are really different, and you have to try and find and understand how can we bridge the issues that the regulators have right now by using these new methods. But so there is right now an EPAA designathon where a lot of ideas were collected, and the first actual meeting takes place at the JRC in March to collect all the information that was submitted. It's not about getting a grant, it's about let's put all the ideas of the people together, collect them, and then collectively try and find a solution how we can really do the transition into application. So that's basically the outline of the landscape. So what does this have to do with SCAT? So SCAT is having different tasks. On the one hand, we do research funding of founding universities. So this is basically the University of Genève. This is where the foundation is, the home of the foundation of SCAT is. Then the University of Lausanne, ETH in Zurich, Fachhochschule Northwest Schweiz, and Uni Basel. And so the idea is that we really channel the research towards regulatory application. And the main topics of the SCAT have been now, and also for the next program, neurotoxicity and endocrine disruption. And so this is why, where I'm coming from, I'm really fitting to the research goals of SCAT. Then we participate in education in the Master of Drug Science and the postgraduate studies in toxicology in Genève for also teaching these new methods and we're actually hosting the first master student this year. Then we interact with the Swiss agency. This is a very important part of SCAT to inform them about what's going on in the outside international world. We work with international agencies, so we are in different OECD expert groups, work with ECHA, the FDA, EPA, and also with EFSA, and bringing this home also to the Swiss agencies. And what I would like to do is perform like a Swiss case study on emerging topic of Switzerland, possibly comparing classical and the novel risk assessments and bringing this back to the OECD. And there, what I would be also very much interested in would be to serve also the One Health aspect. So this is from the precision tox from the different species, but there's also an initiative of the taxonomic domain of applicability from the US EPA, and the idea goes into this one substance, one assessment, one health. And so I'm hoping that I can also interact with the Eawag and the possibly also the Ecotox Center, and that maybe we can do something together towards this direction by involving the Swiss agency and bringing it to the OECD level. So that has been it. I hope I met your expectations. I think it's time for change, and I would very much like also to Switzerland to contribute to the EU roadmap for phasing out animal testing. And I'm looking forward to your questions.